I'm going to be doing a session on biosafety and chemical safety, talking about levels and requirements. So this is part of kind of research oversight. Oh, you may or may not see my closed caption, but I do that. So you'll see my closed caption. So my name is David Sanchez. I'm an associate professor here in the College of Pharmacy. I'm also the chair of the Institutional Biosafety Committee for Western University of Health Sciences. So I've been the chair for about 9 years. So my job is to oversight our research here at Western U to determine two things like, is it being done according to federal and local guidelines, as well as is it being done safely and in a responsible way to protect yourselves as researchers, as well as our local community. And you can imagine, because biohazards come into effect, that's an important thing. So as we're going through, if you have questions, there's plenty of time to talk about stuff. So please let me know. We'll go ahead and talk. And I'll ask some questions here and there. So don't worry, I'll reach out to you all and see what you all think about different things. So, but let me see if I can see it again. Okay. All right. So we're going to have a few learning objectives here. They're pretty straightforward. We're going to talk about the word biohazard. You all probably have a term in your head for that. We'll talk a little bit more about exactly where we're going. We'll talk about biosafety levels, BSL 1 through 4. Again, you probably know about these ideas, but we'll make them a little bit more articulated so you can understand. We're going to talk about the roles and responsibilities of the Institutional Biosafety Committee. Institutional biosafety committees exist all over the place, and we'll see how they exist in other words that you probably have some understanding of, including the institutional, the IRB, including the IRB, the IACUC, stuff like that. And we'll also recognize some examples of research requiring IBC oversight, as well as the ones that require a little bit beyond IBC oversight. And what do we do in those scenarios? So we'll go through them. So how many of you all know what a biohazard is? Could recognize one? Does anybody know? How would you recognize a biohazard? Anybody ever seen anything that makes them recognize that? Anybody walking around see anything?
The symbols?
Yeah, exactly. So a biohazard symbol. So even if you don't know what it is or there, you should see the symbol. So for example, this, right? So hopefully you've all seen that little red biohazard symbol with the little like circles and things like that. That's the classical universal biohazard symbol. So what is a biohazard? It's an agent harmful to humans, animals, or the environment. And we have to think about that. As much as we like to do our research, you have to think about, is it safe to the humans, us, other humans, the animals, as well as the environment, which is going to be important for us to talk about. They could be biological hazards, they could be chemical hazards, or they could be recombinant DNA. So, and that's an interesting thing that we'll come to. Recombinant DNA, we think of innocuously, like if you think about like in research, you think about these are, plasmids and stuff like that, but they also present a biohazard. And then some examples include HIV, which is a straight like understanding that that's a biohazard because the virus that caused disease. E. coli, we know we have E. coli all over ourselves. We have it all over the place, but how is it important? But more importantly, now we have a lot more important fundamental ideas that are coming out. For example, what about CRISPR edited organisms? Are they biohazards? Or if you're introducing a lentiviral vector that changed the genome of the individual or the organism, is that a biohazard then? So I think these are important questions that are starting to come up and develop in there. So as it turns out, biohazard for us is anything that changes or impacts the normal environmental biology that's around us. So when we classify biohazards, we often use this classification called biosafety levels. And so you may have heard about these BSL 1, BSL 2, 3, 4. There are 4 biosafety levels that exist. And they have increasing level of necessary containment of the biohazard, as well as protection for the individuals. So here we have BSL 1, 2, 3, 4. As you go through, they become a little bit more increasing in the amounts of what are called PPEs, as well as safety equipment and facility design. So you might imagine whenever you go into a research lab, there are certain levels of this biosafety that exist. So if you've gone to a research lab ever, or even a regular teaching lab, almost all of them are BSL-1. BSL-1 has no defined containment. You know exactly what you're doing. So for example, you could be working with little E. coli that are growing in a plate and you're just going to spread them. So remember, like in microbiology classes or in intro bio classes, you put bacteria on a plate and you looked at it and that was it. That's BSL-1. The agents that you're using in that scenario that are present have minimal potential hazard, and they're not really hazardous to the people using them or the environment. So, and we'll talk about the autoclave later, but what that means is that if you inadvertently contaminate yourself, you're not likely to get super sick. Maybe you'll feel like not good, but you won't be like sick. In addition, if you inadvertently take some of that sample out and go to the environment and drop it somewhere, it's not likely to cause any environmental impact. So it's interesting because that's a little bit different as you move to BSL-2. On our campus, most of our laboratories are BSL-1. However, a lot of our campus laboratories have additional BSL-2 facilities. These ones are ones that have moderate risk, and there's a potential for a disease depending on what you're doing. So some organisms that you can use in a BSL-2 are influenza virus, HIV virus, different bacterial infections. We have several laboratories on campus that do this type of work directly. And these are agents associated with human disease and pose moderate hazard to the personnel and the environment. Okay, let's think about this. So what's the difference really? The difference is in BSL-1, they're not going to get you sick. In BSL-2, they could get you sick. So for example, let's think about like a person. How could a person get sick in a BSL-2 environment? Anybody think like there's some example organisms? Anybody think any of them could get you sick? How would that happen?
I'm thinking, yeah, maybe if you like come in contact with mucous membranes.
Exactly. Simple. If you inadvertently expose yourself to them, simple stuff. And you mentioned a really important thing, the mucous membranes, right? You know, our hands, we always worry about like making sure we have gloves and stuff like that, but our hands, for the most part, as long as we have intact skin, are not going to have too much problems. The mucous membranes are exactly what you're pointing out, because that's key. If you inadvertently have an aerosol, so for example, have you ever dropped like a soup or something? Has anybody ever dropped a bowl of soup? Does it just go flat, plump? No. When it goes down, it goes flat, frunk, right? And that flat frunk is actually the aerosols that are going. You all of a sudden, when you do that, you can smell the soup a lot. You smell the soup, you smell the food. That's because a lot of it went to aerosol. That's exactly what happens if you have a sample in BSL-2. If you inadvertently drop it, and that's often happens, or it pops open, the aerosols go out, and those often will be what leads to contamination and potential infection for the individual. I'm glad you mentioned it because that's really the key. Our hands, usually we have PPEs, usually we have gloves, but it's exposure to our mucous membranes, our mouth, nose, our eyes. Those type of things are really key. Those provide a route of infection. So we have to have some type of controls, and we'll talk about those in a minute. How do you protect us? BSL-3 and BSL-4 are a little bit different. BSL-3, we do have laboratories at Western U that can protect for a BSL-3 environment. We have two spaces on campus that can do that. Neither of them is used currently as a BSL-3 environment. As you can imagine, they're actually quite regulated. So BSL-3 has high containment, especially for, as we mentioned, aerosol transmission. So these are actually infectious agents that are caused primarily by aerosol transmission and are potentially very serious diseases. For example, the first one there is tuberculosis. Tuberculosis, some of the more high pathogenicity influenzas, if you get exposed to those, the person will get sick. And it's not sick like, oh, you're going to go take a couple of days off work. This is sick where you're going to have to go get medical attention. These often will have indigenous or exotic agents, agents that present a potential for aerosol transmission, and agents causing serious and potentially lethal disease. In here, we don't just have our environment contained, we have something called a pass-through autoclave. What that means is that all your waste, all your extra stuff has to be sterilized before it leaves the laboratory environment. That's when you can imagine it's a more complicated laboratory. We have a capacity. So if you ever in certain laboratories, there's some that exist that are set up that way, but we don't currently have that type of laboratory research going on. Now, the last one, BSL-4, is our maximum containment ones. We don't have that at Western U. We rarely have them at localized big, close to big cities. These are ones that are specialized laboratories. These ones are the ones that are either run by very high-level infectious agents research that are looking at for complicated diseases, or they're run by the military because they're very dangerous. Example of that one is Ebola. So we don't have Ebola work going on at Western U, thankfully. So we don't want to let that out. But that's because the potential for transmission is really high. Tuberculosis will get you sick, but Ebola will get you sick and quickly spread to other people to get them sick if you let it out. That's the difference. Not only that, you have to have lots of controls, and we'll talk about those in a minute. So hopefully you see like BSL 1, 2, 3, 4, you can see the spectrum of low stress where you are doing it in your labs to high level, super trained people that are dealing with infections that could cause outbreaks or pandemics. So any questions about that before we go a little bit deeper dive? So we're going to talk about the logistics here. Any questions at this point? All right, let's move on and get a little bit more technical about these ones. All right, so let's talk first about BSL-1, which has low-risk microbes up to BSL-4. Oops. So I put a little diagram. It's a little blurry. I got it from the CDC website, so they're a little blurry, but you get the idea. So BSL-1 uses standard laboratory practices. So you see the person here has a little A, they're moving chemicals around and stuff like that. So you can work on an open lab bench or a table. You have to have your personal protective equipment. So you all, if you're doing any research in a laboratory setting, will have personal protective equipment. Oops, the chat thing. Oh, I'm sorry that someone had given me an answer in the chat. I didn't see the chat till right now. Thanks. I'll monitor that now. So if you are working in a research lab or if you're going to work in the healthcare field, you know PPEs. Personal protective equipment protects you. Lab coats, gloves, potentially eye protection as you need it. And all their facilities that are doing BSL-1 work, any basic research, must have a sink available for hand washing. And you must separate that from the rest of the facility. So if you go to your research lab, you have to enter a door, right? So if you have a research lab or you're doing any research space or you're doing any clinical research or you're doing anything, there has to be a door. And you might say, that's crazy. Of course there's a door. It's a lab. But a lot of times people will design facilities and have open space and stuff like that and realize you can't have random people from the community just walk into your lab space without training. So that's important. You have to have all your PPEs on. So one important thing about research in the biosafety categories is you always have to have PPEs on. So I assume if you're doing any type of research in clinical or wet lab research, you understand you have to have your PPEs. Basics of BSL-1. As you progress through the BSL charts, you have to have increasing amounts of extra stuff. So let's move on. Oops. Oh. Important things about personal protective equipments. You need minimum PPEs for all work, and you need additional PPEs for particular experiments. So for example, if you need safety glasses, if there's going to be a break and you potentially could get stuff in your eye, or safety goggles if you're going to have splashes. Again, splashes are important because of the mucous membrane that was brought up earlier. The one thing also that we talk about is, and you had basic lab classes, you know this. Whenever you're working in a research lab, you have to have long pants or equivalent and closed-toed shoes. We don't mention these enough because we always think, oh, our lab coat and gloves ready to go. But especially in the summer in research labs, you'll notice, it's not fun to come in with your flip-flops and shorts straight from the beach and have stuff spill on you. So it's really important to think about that. If you're talking to people and noticing that, feel empowered to tell them, hey, maybe you should put some pants on because you don't want to spill onto your shorts and get like hurt. And you'd be surprised how easy that is to protect someone to have simple PPEs on. Any questions about PPEs? Hopefully, if you're working in a research lab or a clinical lab, you have access to PPEs. If you don't and you need a lab coat, just send me an email. I'll get you an email. I'll get you a lab coat for where we're at. So the best thing about our lab coats at Western U, they are managed by an outside contractor. So you should have super access to them, especially if they get dirty. You should be able to get them cleaned out right away. All right, so let's talk about BSL-2 in action. So a couple of things. So if you go to a research lab, some of the labs you can just walk in and kind of meet people. In a BSL-2 area, that area should be restricted. So when work is being conducted, the door should be closed. You shouldn't have people around like hanging around, walking around. You should be kind of focused and be able to do things. You have important PPEs. So the A PPE, this person has lab coat and gloves on this A. They also have a little splash guard. Usually and we'll talk about this in a second, our biosafety cabinets have a splash guard, so you don't really spill things on you. You also have an autoclave to dispose of things. You know what we do at Western U? So instead of having autoclaves everywhere, we take it to an outside contractor who does all of our autoclaving for us. So for Western U, we're a special scenario where we do all of it, so we don't have multiple autoclaves all over the place. You should have a sink and you should have an eye wash. If you work in a research lab or a clinical lab, I'll give you a homework today. Go find out where your local eye wash is. So if you work in a clinical lab or research lab, ask, hey, where's the eye wash? And you might say, oh, well, don't need an eye wash, but I like my eyes. I like being able to see. So it's good to know where your eye wash is. As much as you might say, I'm never going to get anything if you use gloves and anything, accidents happen. It's always good to know where your eye wash is. Chemical showers exist too if you need to know it. But if you don't know where the eye wash is, go and ask your local lab people, where's the eye wash? So this is a regular BSL-2 lab. It has a biosafety cabinet. I think I'm going to talk about that. Yeah. Let's talk about what a biosafety cabinet is. So a biosafety cabinet is the primary engineering control used to protect personnel against biohazards or infectious agents. And it helps maintain quality control of the material being worked with as it filters both the inflow and exhaust air. Is anybody working with a biosafety cabinet right now? Anybody ever seen one before or used one? Oftentimes we use this to separate ourselves from potentially dangerous things as well as keep them safe. So what's cool about this, so you see you see the person sitting here, right? You notice that they're sitting out here with their normal PPEs, but the work they're doing is behind this like kind of glass plastic thing. And then they have a little internal flat bench where they're working on things. The biosafety cabinet is key because it's separating the two, the individual and the items they're working with. So let's see how it works. So there, you see her hands here, and these are the hands that she's working with on the other side. Inside here, there's contaminants on the outside and there's contaminants potentially on the inside. What happens is the biosafety cabinet, through that front grill, the little dots there, sucks in the air. The air then cycles around the entire biosafety cabinet back and gets HEPA filtered coming down. Everybody know what a HEPA filter means? Anybody heard of HEPA filter before? Good question. Good question. I'll get back to that question in 2 seconds. So a HEPA filter is a way to just separate out the air so it's 100% sterile air. So there's no bad things. Now, the great question in the chat is this the same thing as a fume hood? So no, it's the kind of the opposite. So a, so you've ever used a chemical hood, right? So let's say you took organic chemistry or stuff like that, and you use a chemical hood when you're something smelly you're working with, right? You don't want to smell it. You put it in there. What happens? All the fumes go straight up. Chemical hoods or fume hoods often will take that away. Biosafety cabinets, on the other hand, will push the air down because they're sterilizing it and the air is coming down to do two things: keep everything sterile, as well as clean the air from things don't attack you as the user, and things don't mess up the experiment as the experiment. So that's a really important distinction. Some biosafety cabinets have the fume hood as part of them, but they're separate ideas. So if you go into a laboratory and see a fume hood or a chemical hood, it's not the same as a biosafety cabinet. So I'm really glad you asked that distinction, because it's really an important thing, because some people don't know. And you'll go inside and they won't know, and they'll be doing one or the other. The biosafety cabinet protects you and protects the work that you're doing. So it's really important. If you're dealing with bacteria or cells that you want to keep sterile and not contaminated with outside fungus and bacteria, biosafety cabinet is the way to go. If you're dealing with anything pathogenic, so you don't get infected, the biosafety cabinet is the way to go. Because you see the little virus in the middle of the biosafety cabinet picture, little orangey virus, it can't go out. It gets sucked back into all those grates and all those grills marks, because it's always being sucked in and HEPA filtered out. So those things are constantly being cleared. At Western U, we every year we go and check them to make sure that they're doing exactly what we want to protect the user. We clean them out. We do all that stuff. So every year we did that. I think in July we do it. So we're at the end of the cycle. We're going to do it again to make sure they're always safe for the user. I can't overstate how important a biosafety cabinet is to protect you from a biohazard. This is our key engineering control, especially at Western U, because we have so many different labs. So if you're in a lab that's dealing with something, any type of biohazard, you should have access to a biosafety cabinet. If you don't know, if you've never seen one and are curious about one, send me an email. I can happily show you what it's all about. If you're around. If not, is somebody at Comp Northwest here?
Yeah, that's me.
Okay. Yeah. I don't know. So if you don't do anything, I can connect you with somebody up there that can show you what a biohazard cabinet is. So
Okay, I'm actually doing clinical research, so I'm a little
Okay.
out of this. Yeah, but
Yes.
thank you.
No, so I'm saying if you don't have access to that or want to see one, I can connect you with some of the people up there and they can show you what it is about. So
Okay, thank you.
yeah. So, and that's the point. So I'm the IBC chair for Western U, both campuses. So I oversee the research that's going on here as well as the research over there in terms of biohazards. So I'm glad to hear people on both campuses. Okay, let's talk about BSL-3 action. So this one's a little bit more complicated. This person's a little bit more PPE than we're expecting, right? So someone that's working with things like tuberculosis or high infectious viruses is going to have a little bit more PPEs. These are going to oftentimes have respirators. Why do you need a respirator for this research? Especially if things like tuberculosis. What's the respirator for?
Protecting against aerosols.
Yeah. So what's going to happen is that if you're dealing, so for example, let's say you're doing everything in the biosafety cabinet, you're doing everything perfectly, and you step out and you slip by accident, people slip, and it drops. And remember the soup, plump, flump. The bacteria is in the air. You don't want to breathe in bacteria. The respirator sterilizes the air as it's going in and filters it out, so you would be totally safe. You also have more advanced PPEs. These ones are covering. They're often disposable, or we have special types of ones. We take all the PPEs out and dispose of them and let the the room circulate the air. Our BSL-3s have a separated room that has airflow separated and HEPA filtered out. So that room gets air HEPA. So it's like you're in a biosafety cabinet because the air inside the room is being filtered out. You also have hands-free sink and eye wash available. The key here is hands-free. Because if you're dealing with things that are potentially infectious, you don't want to be touching a sink. Because remember, and this is important for you all, healthcare people realize this, when you're using a sink, that's a really great place to cause transmission of contaminated items, right? The sink handles are fomites. You basically touch them and then you wash your hands and they're still dirty from original and you touch them again. So hands-free is really important. So you might think, oh, well, whatever. It's really key. So always make sure you try to use hands-free when you're dealing with anything like that. The one thing about here is the exhaust air is not recirculated, and we HEPA filter the air when it's done. So when you're done working in there, the air is HEPA filtered before it leaves. Why do we HEPA filter the air after you're done with all the work? We HEPA filter the air as it leaves to the environment. Why do we do that? Who are we protecting in that case if we're HEPA filtering air that leaves the lab? We're already left. So who's left? Perfect, the community. So if you HEPA filter the air, so let's say you had a big thing, again, any high infectious influenza, high infectious bacteria, you're done. The air goes out. You don't want, the person walking by the street to get some air doused on them and get tuberculosis, to get high infectious influenza. So we HEPA filter and protect those individuals as well as our individuals. So that's a really key part. All of our BSL-3s on campus have a HEPA filter system on the exhaust air. So even though the air goes straight, so we have our BSL-3s are in HEC and RWC. They go straight up really far, and as they leave, they're HEPA filtered. So it's really clean to keep that our community involved. So perfect. Let's see. The last one is BSL-4. I'm not going to talk about this too much. This person's in a spacesuit. Same issues, right? You don't want to get exposed, and your air intake is totally separate. You also usually will do like showering and stuff like that because you don't want to potentially take anything with you. And you have a lot of what's called medical surveillance. So BSL 2, 3, 4 have medical surveillance. You want to make sure your individuals that are working there understand the risks and understand how to monitor themselves for potential exposure. You can imagine if, let's say you were working, so have you ever seen those movies like Contagion or like Outbreak or stuff like that? Those individuals, they go to places where there's outbreaks. And the first thing that they're taught is if you're exposed to this, these are the signs and symptoms you're going to start feeling. Someone in a BSL 2, 3, 4 should have some understanding that, if there's some big agent that you're going to use, for example, if you're going to use an infectious agent, you should know what the signs, symptoms are for potential exposure. And that's really important because, for example, let's say HIV. If you potentially had an exposure to HIV, like you said, oh, I had a needle stick somewhere, you would know, oh, I can go get PEP and I would be protected. Or if you started getting sick and you say, oh, I'm starting to feel like a cough and runny nose or stuff like that. Oh, I may have gotten a respiratory infection. I can go get medical care. That's really key. And you all you all are healthcare, so you understand interventions early in the course of disease are the best approach. So that's why you always alert your users what to do. Okay. So, now the question is, so how do we oversee all this research is done safely? Because you might get kind of a little concerned, like this sounds like a lot of complicated stuff. I don't want to get sick. I don't want people to get sick. So we have an institutional biosafety committee. They are an NIH mandated oversight of biohazardous research. So they are mandated by the National Institutes of Health. And they include scientists, biosafety officers, community members. So I like this diagram because you may, I think if you talk to Jennifer Baker, you may have heard her. She talked about, IRB access, IACOC access, all that city training. All of that works together with the community. So we work all together. So I know all the people that work on these different oversights, and we all work together to make sure everything is done appropriately. So the things that IBC oversees are recombinant or synthetic nucleic acids, human gene transfer, select agents, BSL2 plus agents, and we also monitor environmental and community risk assessment. The cool thing was in the middle, we also monitor plant biosafety, which is important for agriculture. We also connect with IACOC to work on animal welfare because we want to keep our animal welfare of our environment. And of course, if there's any human research, we work with IRB to determine what's the best way to protect people. From our standpoint, IBC's real goal is to protect the users and the community. So we always want to make sure that they have access to those things. So here's the Western U IBC. So if you ever questions about IBC, you can just email ibc@westernu.edu. We are underneath the Office of Research and Biotechnology, Office of Research Regulatory Affairs. And so here's how it works. The IBC is composed of several different members. The chair is me. We have scientific members. I'm also a scientific member because I am a virologist, so I bring a virology. But we have people who are interested in bacteriology. We have people who are interested in more complicated things like biotechnology. We have someone who's representing animal care. So our veterinarian on campus will come in and give feedback. We have an outside biosafety officer. So she's a really super experienced person that really understands biosafety at a very technical level. So how to protect people, the regulations that are required. We also have environmental health and safety members. So people on campus that their job is to make sure that you're all safe from a more environmental site on campus. So do you have the right infrastructure, the right buildings, the right stuff like that? But I think the most important people are our community members. Whenever you have oversight groups, the most important people are the community members. What they do is they bring an outside perspective. What do you think the questions a community member would bring would be? What do you think they would ask?
I'd imagine the most basic questions about their own safety, the safety of the environment.
So you said you had 3 points that are really good there. They first of all ask the basic questions. Because you know what happens with scientists when they write about their research? They write it crazy complicated. And sometimes it's so complicated you can't understand what's going on. So they ask really basic questions. They also say, how are they going to be safe? If they're a community member that walks by Western U, how are they safe? And how is the rest of the community safe? So you know what? One of our community members, he's amazing. He used to be the healthcare director for Pomona Unified School District. And you know what he asks? He asks us, if something goes wrong, could this become a front page of the LA Times story? And he asks us that for a lot of things. Because, our researchers are like, oh, of course, it's easy. I'll take care of it. But the community members ask the really hard questions because they want to protect not only the Western U, but they also want to protect the community. And the cool thing is they're not affiliated with Western U. So they don't care if they say something really like alarming to us and tell you have to fix this because they're not paid by us. So whenever you deal with these advisory boards or these oversight committees, especially as you go forward, there's always community members there that are not in conflict. Because theoretically, outside perspective is I'm in conflict because I work for Western U, but the community members are not. They are recruited from outside. They do it totally voluntary, and they're the best volunteers you've ever met because they want to keep us safe. So we have 3 community members. What the person I mentioned was retired. The other person is the current school district health person, and the other one is a regulatory affairs person from one of the biotech companies. But he lives here with his family and stuff like that. So it's kind of like everybody brings a perspective. Oops. So little details there. So how does this? Sorry. All right. Does your research have an IBC approval? So if your research involves any of these things, it likely has an IBC approval. If you deal with human cells or cell lines, if you deal with recombinant DNA, infectious agents, the CDC select agents, which are the ones that the Centers for Disease Control, the zoonotic pathogens, which are ones that could infect animals, or transgenic animals, if you do basic research with transgenes, we have approval for it probably. We want to make sure all these things are done safely and appropriately. And then what we do is we do it in partnership. So when we get an IBC protocol, we review this. So this is actually more technical for my side of how it happens. But we have an administrative review. We do our city training. So you've all done city training, right? That's exactly what we want. Make sure everybody's done the city training already. And then we do a lot of working with each other. So I work a lot with the PI to make sure all the paperwork is done in terms of done right. Jennifer Baker does a lot of the administrative paperwork. And then we work with the other regulatory bodies, like the Institutional Review Board, the IACOC, the Radiation Safety, DIRC, SCRO, and EH&S. And I'm going to talk about DIRC and SCRO in a minute. Then we have a full review. So it's a big process of us making sure everybody's in partnership and working together. I like it because it means we're all doing stuff together. So this happens. So when we have this question, we mentioned like, well, are you doing it safely? But another question is, should you even do it? Is there research you can imagine you shouldn't do? Has anybody ever thought of research you shouldn't do? You can type in the chat if you want to brainstorm.
Can you clarify the question? Sorry.
Yeah, is there a research, let's thinking about this biohazard research, anything, would there research you wouldn't do? Felicia mentions research that harms others. That's an important one. So
Maybe
if somebody harms,
something like a very rare disease that's highly contagious.
yeah, so working on those ones, you potentially could make it like different. So has anybody ever heard of gain of function research? This one came out, so at the beginning of all the COVID stuff, people brought this idea up a lot. If you're doing research on a very contagious disease, what happens if you make it more contagious? Is that good research or bad research? So gain of function research is a really complicated one. So the question is, should all research be conducted? So let's talk about gain of function or infectious disease stuff first. So we have something called dual use research of concern. And I'm going to read this because I got this straight from the government, what to say. So it's a small subset of life science research that, based on our current understanding, can be reasonably anticipated to provide knowledge, information, products, or technologies that could be directly misapplied to pose a significant threat with broad potential consequences to public health, safety, agriculture, or others, or even national security. There are some examples down there. So different dual use research includes, what have you made really much better nuclear energy or radiology that could be turned to nuclear weapons? What have you made a much more advanced virus that you could study, but it could be a bioweapon? What if you had genetic enhancements that could change things and change people? Or nanotechnology that could deliver small drops that could be made into weird weapons? Weapon. But anything that could potentially hurt somebody. So I think those are the two things. So harm to others, but potentially harm to a lot of things. And you might say, okay, I understand the biohazard one. COVID came out where the idea was SARS-CoV-2 a laboratory made virus. Most people don't feel that's true, but that was a big question that came out at the beginning of COVID. Was it a virus that was made in a lab that got accidentally released? This is the type of research Dirk oversees. Should the research be done? And if it's done, would it be a danger? And you might imagine, wow, this is complicated, but that's why we have it. We have it in place to oversee that, to protect. If the research is too dangerous, we won't let it happen. So that's an important thing for you all to realize as, people that care about our community and care about the health of individuals. We will make sure research doesn't impact our society. And even if the research can progress forward, there are other ways. So for example, I'm an editor of a journal, and when we have research presented to us to publish, that is a Dirk research, we go and ask the university, do you have approval for their research? And if they don't, we won't publish it, because we don't want people to know how to use dangerous research. So we have a research oversight like that. So a little bit different. Any questions about Dirk? Because it's a little complicated and we never think about it, but we do oversee complicated research. You know what was an interesting version of Dirk that I was told? Oh, how long has this been in place? Since the mid 2000s. So like before 2010. And the reason it started to come in place was because we started getting concerns about bioterrorism. And we wanted to be sure that we had oversight in our laboratory. Gain-of-function research with influenza was the other one that triggered this. We didn't want to have pandemic influenza. So a lot of people that did influenza research put a lot of feedback onto there. And every year we update it to make sure it's more in place. So things like SARS-CoV-2 have impacted that and going forward. So as soon as we got it, Western U got our setup for that. So we're pretty upset. Interesting enough, we have zero Dirk research at Western U. So don't worry. I think the bigger universities have that. So bigger healthcare universities have this. Now the other one's a little interesting. So I think I put it backwards. The other one is SCRO. These are stem cell research oversight. This one reviews the scientific merit and ethical justification for proposed research involving human embryos and embryonic stem cells. This one's way more complicated because during the Bush administration, the second Bush administration, they put a lot of mandates and controls over people using stem cells from embryos. Because of that, we have a lot of laws in place to control the use of stem cells from embryos. Of course, this has different justifications on what's important and what's not, but the idea is that we have a mechanism in place to protect us from the misuse of human stem cells and human embryonic stem cells. A lot of people use these type of cells because they're very good, especially now. If you've ever heard of induced pluripotent stem cells, iPSCs. iPSCs are stem cells you can degenerate back to a stem cell and then generate into any type of cell. So you take the cell, generate it back to a stem cell, and generate like to skin or to lungs or to kidneys. You can do all this type of stuff, but should you do it? And how do you do it? Can you genetically modify it? How much of an organism can you make? These are important questions that we're not ready to understand because nobody does them. But in your future, when you're like a seasoned healthcare provider overseeing departments, one day you're going to have these type of questions come to your department when you're going to say, oh, this person is building an entire organism out of stem cells. What do we do? So we're at a very early stage of this, but over time, people are going to have these questions. The other question that comes out is a purely, ethical one. Should you even use stem cells that are from embryos? These are important questions that people bring up. Our community has lots of discussions on that, and we're not at a set rate to understand, can you do research on that? So I think a lot of questions come out here. They're very poignant questions. So when you talk to people, some people get very passionate about different ideas on here. So it's a very important thing to realize there are people that kind of go through these questions. And this one's been in place since the early 2000s too. So it's not the same question, but I'm going to answer it again. So I think that's all. So thanks everybody for listening. Do you all have any questions about anything we talked about? Or again, I'm going to go back. I'll put my sanchezd@westernu.edu. You can ask me any of these questions or you have follow-up. Any questions otherwise you might have? Hopefully this gave you a little bit of feedback so you can understand some of the things going on. No questions in chat. Sounds like everybody's happy. Well, there's my email address. I'm done. Thanks everybody for your time. Natalie, I don't think there's anything else I have to do, right? I don't have to.
No, that's all. Thank you so much.
Thank you all. It was good talking to you all. If you have questions, just let me know. Aaliyah works in my lab, so if you need to get in contact with me and lost my email, just talk to her and you can contact me that way. So nice meeting you all.
Thank you.
Oh my gosh, they went fast.